Okay, I think I'm ready to go. Too many things to toggle through. I think I'm getting it figured out. Good. All right. We got pretty close to wrapping up the worms. We still have some fungi we want to think about in chapter 5. So about to go away. There we go. I left off with the guinea worm Monday. And this is something, first up, I think is pretty awesome. This is a parasite that we feel like we are super close to having this eradicated, we might say. Human infections are almost non-existent now. A lot of the problem with this parasite really is kind of dirty water. Drinking water that has some aquatic invertebrates, copepods, accidentally consuming some of these that harbor the parasite is how people become infected. So recognizing that that's how the infection occurs, going into areas where the parasite is common, endemic, providing education about clean water, filtering the water or boiling the water one way or another to try to ensure you're not just drinking straight from the stream, has dramatically reduced the amounts of human infections. A couple of countries in Africa still harboring it, where we see a small handful of people that have been infected. With adequate treatment, we can cure these individuals. So we feel like we've about annihilated it. But did we? What happens to something when what it once was infecting no longer is accessible? Do you think it's just going to flat out die? Why would it? Uh, it's just going to shift to somebody else. There are all sorts of different types of infectious disease that have jumped ship, that have went from somebody, some animal into some person, or vice versa. All right. Multiple opportunities for things like that to happen. And we will think about some of those as we progress. So, jumping ship, we are seeing now humans not that big of a concern, but dogs definitely are. And if you have dogs and you happen to take your dog camping, why wouldn't you? If you happen to do that, the first thing your dog probably does is jump into the stream and drink a bunch of water, right? Uh, that's pretty stinking common. So the potential for this to shift from human issues to veterinary issues is really kind of the transition that we are witnessing right now. You can't tell your dog, don't drink out of the stream, right? Probably not going to listen. All right. So that's why Fido's over here, because they're truly becoming more of the problem. All right. So typically we think about, and this image from the CDC, they've got awesome life cycle images, more than just parasites. When we think about all sorts of infectious disease, but for the parasites, really some pretty cool stuff. We've seen a few, we'll see more of them. So the unfiltered water is what we typically think about, but there are also opportunities where we would call these guys parentenic hosts, a transport host, something that that parasite doesn't need to be in to complete its development. It just hangs out and then moves hopefully to the next host. So eating something that's not cooked properly, raw fish or frog, something that has fed upon these copepods and then we feed upon them, also could set the stage for infection. So this is something that you drink that copepod, the larval stage within it gets degraded out as it's moving through your gut and into the intestines. All right. They find a mate, pair up, mate, and the male, like I said with most of them, dies shortly thereafter. As we look at this image, these are a type of roundworm. So the male right here, just little, all right, and the female gigantic, all right. Shortly after copulation, she kind of starts this migration process, and it takes her about a year to complete this process. And in this whole time in transit, these eggs are being developed within her, and she's growing to about a meter in length. That's pretty stinking gigantic, all right? And as she becomes basically ready to go with those mature eggs, it's time to start releasing these progeny, all right? So what happens, and it's typically the foot, but the ulcer can appear elsewhere, but usually it's the foot, all right? So we're looking at the back. We're looking at the ankle right here, Achilles tendon. All right. That seems like a super painful place. What are you going to do if you've got a sore on your foot? Probably soak it, right? I bet you that's what you'd like to do because that sucker hurts. So where are you going to soak it? Right in the stream. That's also where I drink my water. It's where I bathe, probably where I go to the bathroom, right? All of these things could be happening right in this pool of water, and you put your foot in there and soak it. That's exactly what that worm is hoping to have happen. Again, if hopes could be, right? So it wants your foot to go to the water because once that happens, the juveniles start getting deposited out. I'm going to release my young, all right? And you're going to do this repeatedly, right? So as these larvae get released out of that worm that's emerging through your foot, they swim around, and these little copepods are like, sweet dinner, and they eat them. They become infected, and the cycle is completed. All right. So We can see a matchstick up here. This is a common mechanism to rid yourself of the worm. When she pokes through and starts laying those eggs, you can slowly twist her around this stick. She's a meter long. You can't just pull her right out. Over the course of a couple of weeks, you slowly turn it out. All right. Impressive and also a bit disturbing. All right. There are numerous accounts of this going back to antiquity with guinea worms. All right. There's biblical accounts of the guinea worm. All right. All sorts of details about it. Scourges upon humanity. Right. And they imagine it's probably pretty accurate. The medical insignia, the staff with the serpents circling around it is really these guys, all right? Not snakes, but worms, all right? Serpents, which is what really we kind of thought these things were like in the first place. When you see, you're like, what is that crazy thing, right? Pretty impressive, all right? Okay, shifting to the fungi. These have a huge amount of variation when we think about fungi. All right. Oftentimes, when I think about fungus, I think about mushrooms. All right. That is a pretty common thing to pop into my mind. I also sometimes think about some container that got pushed to the back of the fridge and forgotten about for months. And then you pull it out, and that sucker's growing all sorts of crazy mold. All right. I feel like I'm a little better with that now. Maybe part of that could be children. I don't know. There's lots of, lots of dirty diapers and gross stuff that kind of hardens you, I feel like, getting puked on and whatever it might be, right? I can be in a microbiology lab and I can open up one of those incubators, and you have all smelled them, I'm sure. And as the semester progresses, we are going to ripen some things up in there that smells really pretty disgusting. Smells like butt, because that's where that stuff's coming from, right? Makes sense. And I expect that. So it typically never phases me as I open that incubator up, and it's also cooking hot as you open it, right? Expect those things. But I open my fridge up and open up one of those Tupperwares and I about want to puke. Right? I'm like, what? I could have eaten that, right? And it makes me a bit nauseous. All right. But why? Right? It shouldn't, but it kind of does. All right. Like I said, I feel a little bit better about some of that now. And I also feel like maybe we aren't keeping things pushed to the back. So the potential for seeing moldy stuff has diminished a bit. All right. So mushrooms or molds, two pretty common things when I think about fungi. All right. When I was an undergraduate microbiology major, bacteria was really what I was thinking about. And as I was starting graduate school, I had an opportunity to work with in a lab that was microbiology oriented, but it wasn't what I was thinking about. It was a bunch of fungi. And initially I was like, is that something that do I want to shift from focusing on bacteria to somebody completely different? And as I looked a little more into it, this type of fungus, pathogenic, really pretty crazy stuff that it can do and how it lives, I was like, that's pretty awesome. And that's where my graduate studies ultimately lent me, thinking about it and how we could detect it through different mechanisms to truly try to get an understanding of the disease process. So We'll get to some of those guys in a second, all right? Initially, when we think about the fungi, they're with molds or our mushrooms, they start off typically in the soils as some sort of filaments, hyphae, individual thread-like strands. If you see a collection of all of that, we call that mycelium. It's just a bunch of hyphae, all right? So the big patch of mold on your bread or on an orange, all of it together is mycelium. Individual pieces are the hyphae, all right? Oftentimes you see some pigments in there. A lot of that stuff are the reproductive structures, the spores that those fungi are generating. We're not showing that on here yet, but you'll see some in a second, all right? So we saw a little bit back some images of fungi, and I mentioned some of the fungi, the hyphae, have individual partitions, cell walls in between them, and we call them septate. And others lack that. They still have individual nuclei, all right? But it looks like it's just a multinucleated thread, all right? No individual, at least visible divisions of these cells, all right? So non-septate or cenocytic versus the septate hyphae threads, all right? Next door, the last image isn't, in my book, the best image. Yeast cells are individual cells that look kind of oval shaped. When they grow on culture media, yeasts look very different than molds, all right? Molds very fuzzy, yeasts almost sort of bacteria-like, but kind of drier looking colonies, all right, typically. So some of the yeasts have the potential in the process of replicating or maybe just living to produce some projections that we call pseudohyphae. But as I see this image, I really am not a big fan of it. It looks a lot like this, which isn't super accurate, all right? So individual little oval cells, these are the yeasts, all right? And they replicate by budding. So they create a new little, we call it a mother cell and a daughter cell, replicates the DNA, and the daughter buds off of the mother cell. Oftentimes the mother is left with multiple scars, bud scars, where her progeny have broken off of her, all right? So some of the fungi, maybe one handful, maybe a few handfuls. Some of these fungi are a little bit unique. Shapeshifters, maybe. They exist oftentimes in the environment at, room temperature sorts of conditions. Environmental conditions lend to the hyphal growth, the filamentous growth, all right, mold that we see. Those molds start generating spores, they're reproductive structures. And if you happen upon these as the spores are being released and you inhale them, now you got problems, all right? Those spores get into your lungs. Now all of a sudden it's a nice warmer than environmental temperatures, nice warm, nutrient-rich environment. And those spores that would settle back down in the environment and grow as a mold now say, sweet, I'm at the buffet. They turn into yeast cells. They start replicating and potentially establish a disease progression within that host. Depends on numerous things as far as will you exhibit any sorts of symptoms or not, all right? So Histoplasma capsulatum is a dimorphic fungus starting off in the environment as a mold and then converting into yeast cells within our bodies, all right, within our lungs, all right? And as they progress, if they truly have the capacity to establish disease, the potential for them to disseminate throughout the body is huge. That may be truly your first hint that you've got something up, all right? You might think initially kind of some chest cold sort of situation, but then it progresses to some skin manifestations or other manifestations on your body as it started to disseminate. And in those circumstances, I mean, they are yeast cells, but I kind of feel like at this point, I think of it like you're sort of molding from the inside out is kind of what's happening as this fungi is coursing through your body. So we can see it within the circulation, the lymphatics, disseminating to the skin, potential for central nervous system involvement, bone involvement. Basically, every body organ has the potential to become impacted. All right. Awesome. Oftentimes, when we think about fungi and we're thinking infectious stuff, the first thing that maybe pops into your mind is it a dimorphic fungus? And in order to figure that out, we cultivate the sample at 25 degrees Celsius and at 37 degrees Celsius. You're going to do, you probably already are doing some of that stuff in the lab and maybe seeing that some of the bacteria give you some different characteristics at these two different temperatures. The dimorphic fungi at 25, you get the mold, and at 37, you get the yeast. You can verify. That's who we're truly looking at, all right? Temperature dependency with that, all right? Pretty impressive. So we're going to mention as we progress through here, and definitely as we get to the tail end with infectious disease, more details about the dimorphic fungi, all right? And with that, there are some geographical kind of separations for some of these. And we are also recognizing as climates are changing, as we are seeing shifts, that the potential for some of these is shifting as well, marching into habitats where they might once have been considered to not exist. We're starting to see some of it. That's a little bit terrifying, all right? Right now, Idaho, we don't think about any of these dimorphic fungi, all right? Coccidioides is one that likes more of an arid sort of habitat. It's seen around the southwestern United States, Southern California. They call it valley fever for the San Joaquin Valley. Lots of issues with that. And that is a fungus that we are seeing moving more north, all right? One that potentially could become a concern, all right? Histoplasma, as we've got the top little inset, is a type of fungi that likes very rich organic material, bird or bat droppings especially, are likely to be sources for the fungi and potentially for us. So we can see up in this attic, some bats, the guano down below, full of this, and then maybe you go up in there and get exposed. We'll touch on more of those as we progress through. I'll tell you some details about blastomyces. That was the dimorphic fungi that I studied, but not right now. All right. So we got a handful of life cycles that we want to look at. And I, again, don't want you to get super bogged down with these details. The idea is to just see some of the similarities with our different types. When it comes to classifying the fungi, when we're thinking our molds, there are a lot of ways that we start evaluating it. One of the first ones is based off of how they are able to reproduce. Some instances, we only know asexual replication, and then that doesn't mean that's all they do. We just maybe haven't figured out the sexual phase of that fungus. There are others that exhibit both. And then there's variations within that that allows us to classify them a little bit further. All right. So first off, asexual or both, and then we start further dividing. All right. Oftentimes, the fungi just hanging out in their environment, breaking down stuff. Oftentimes, we're going to see them really kind of just as asexual fungi, able to replicate by themselves and keep growing. But if the conditions present, if two hyphae happen to be growing very close to one another and they happen to be opposite mating types, we don't call the fungi male and female, all right? It's not like that, but it sort of is, all right? We just say opposite mating types, plus and minus, all right? If you get two opposite mating types that are growing close together, that's what we see right here, positive and negative. Both of these hyphal filaments are haploid, right? 1N. They come together and fuse. And now we've got like sexual recombination, but not. All right. We get a cell that's got what we call it's now called a dikaryon. It's a double nucleated cell. All right. It's not like the fusion of egg and sperm to create a diploid zygote. All right. It's the fusion of these two cells, and the nuclei are within there. All right. So we start off like this, and then we create this 2N. So like kind of like fertilization, all right, to this zygote that's diploid, all right. As soon as this happens, it goes through meiosis to generate haploid spores. And that's a lot of the fuzz that you're seeing on the fungi, all right, are these haploid spores. They get released out. These others in asexual replication also release haploid spores, all right? They just haven't recombined, all right? These in turn go through meiosis. It does give a bit of genetic variation, all right, as we roll through meiosis, but it's not truly the same like sexual recombination, splitting a diploid chromosome in half, or diploid organisms chromosomes in half and recombining that. It's different, all right? Same but different, all right? So that's the idea, all right? And again, multiple ones, and the ways that they form their spores, if we were to visualize them under the microscope, they appear differently, all right, as far as their reproductive structures that they're laying down, all right? So here we can see some hyphae and then the sporangium. This is the sac on the end of that hyphal filament that's going to be full of the spores that have gone through the meiosis, generating those haploid fragments to be bust open and released out, settling down to grow as more hyphae, all right? This is a chunk of bread, all right? I always tell this story. I find it a bit disturbing, all right? There are definitely foodstuffs that you can eat that have mold in them, you know, some delicious super ripe blue cheese, maybe, right? Maybe some tempeh, some soybeans that have been fermented. I tried it once and I'm like, I don't know if I like it, right? Not a huge fan, but I thought I'll give it a shot, right? I had a student, this was in my biology class, and we were talking a little bit about mold. And first off, I feel like when somebody says to me, I have a friend, they are probably talking about themselves. Probably. All right. Not 100% sure, but I feel like that's probably what's up. So he raises his hand and says, I have a friend who likes to get his bread and wait until the bread is moldy, and then he makes his sandwiches. And I was like, huh, that's kind of weird. And I don't know if I would recommend doing that. Why would he do that? I asked. And he said that his friend said he likes the tang that it has, which makes me kind of think of sort of yogurty stuff, like the kind of plain yogurt tang that it has. That's kind of what I'm envisioning in this bread. But I don't know. All right. I recommend not eating moldy bread. All right. Uh, Probably bad news. All right. Here's one more moldy bread thing. We will see it a little bit later. All right. With disease stuff. My gut tells me probably all of us have opened up a loaf of bread and there's been some mold in there, right? Probably all of us have seen it. We've come in contact with it. Maybe you've made yourself a sandwich, you took a bite, and then you look down and you're like, shit, there's mold on this, and you throw it away, right? All of that probably has occurred at some point in your life, all right? There are some people, it's not just regular, normal immune system people, because we wouldn't be here, right? But there are some people whose immune systems are super depleted, all right? They've got all sorts of issues with things potentially causing infections. Could be maybe someone that's progressed to AIDS, could be some genetic situations where they're unable, whatever it might be, all right? There are circumstances where simply opening that loaf of moldy bread and you accidentally inhale some of these spores, that mold can make its way into your body, get into your brain, and kill you in about 48 hours. All right. Mucormycosis, uh, terrifying. All right. Now I think you're not going to be eating moldy bread anymore, right? You're like, first, he had me at the tangy flavor. I was pretty sure I wanted to try some, right? And then he tells me I could die, right? Uh, the odds are you're not going to die, all right? But you could, all right? And that's a bit terrifying to me. All right. So this is just a blow up of our bread mold, and we can sort of see little white strands, the hyphae, but the black speckles, those are the fruiting bodies, the sporangium for this bread mold. All right. And another thing, when you see a bit of mold on the bread, it's super likely that the hyphal filaments are considerably deeper into that bread. So simply flipping that one slice off and eating the rest of it, probably not wise. All right. Uh, Probably not. All right. Um, if we have cheese that gets a little bit of mold on it, we just chop that chunk off, throw it out. We eat the rest of the cheese because it's not like that. At least I don't think. All right. Uh, but bread, that's got to go. All right. My last bit with bread. All right. And I won't ramble on about it. I have recognized, and it all has to do with kids. All right. When my wife was pregnant with our first child, first off, I thought, holy cow, she's going to want to eat all sorts of stuff. That wasn't the case. So I bring home some bread, open it up, and she walks in the kitchen and she's like, oh, that's moldy, right? And I'm looking at it. I see absolutely nothing. I smell it. It smells like bread, but she smelled it, right? She's like, I'm not eating it, right? I ate many sandwiches, all right? I didn't smell anything. I didn't see anything. Once she became pregnant, I felt like her sense of smell was like all of the sudden her superpower, right? Maybe she wouldn't have classified it as a superpower, but a superpower. The ability to smell stuff, I was just like, what? Right? That makes no sense. But then I thought about it and I'm like, it makes perfect sense. She's got this bun in the oven, right? Her body does not want her to eat something that's potentially going to cause harm. It's like, that's bad news, right? I spouted that stuff off. And I had a student raise her hand and she agreed. And she's like, yeah, my sense of smell was considerably heightened. My husband stunk. And I was like, what? She's like, I don't know. She's like, he didn't, it was no different, but I got pregnant and I'm just like, whoa, you smell weird, right? Uh, Weird, right? She's like, for the first little bit, I'm like, dude, you got to sleep on the couch, right? I'm like, wow, at least it wasn't like that, right? Super weird. And maybe, ladies, maybe maybe you agree. I don't know really what's up with it, but I feel like it's protective, right? Makes sense. Pack back. I feel it. All right. So this is just a couple of different examples of some of the spores that we can see. And then yeasts. This is a better image. So a bunch of little oval shapes. All right. These are the individual yeasts. All of our fungi, the molds or the yeasts, their cell walls are made up of chitin. So polysaccharide that makes it pretty tough for us, indigestible. Not that we want to be eating these guys, but eating mushrooms on your pizza, right? The chitin in their cell walls is basically fiber. We don't digest it, but it provides some roughage. All right. So it gives them some protection in their cell walls. And some of our yeasts, really thick cell walls that you can see through the microscope, and some kind of thinner. And you can sort of differentiate a little bit based off of that. So here they're all kind of clustered up together. There are also the potentials for a few, maybe pseudo hyphae, little elongations or projections. And some of that can serve to kind of anchor themselves in the body in between some epithelial cells and kind of tether themselves, a virulence factor, all right, as a potential. It also may be in the process of replicating, where you see some of those pseudo hyphae, but a little further investigation, you're like, it's all just yeast cells. All right. These ascospores, the individual peas in the pod, the ascus, that's what I think of when I see this. And there's a whole bunch of them, but they look like a microscopic image of some peas is what I think about. All right. So different fungi producing these, and each of these opening up like a pea pod kind of, and the spores released out is the idea. So similar idea with this Ascomycetes, the spores, the asexual and sexual reproduction, again, fusion of the opposite mating types, and then producing the peas in the pod, if you will. All right. And we're not going to bog down with all of that stuff. The Basidiomycetes, Basidiomycota, these are the like true mushrooms. All right. And in the soil, we see the hypofilaments, the spores, and then we can see the basidiocarp. Sorry, they're releasing the spores. I started going both ways, but it's a circle. All right. So to our fruiting bodies, oftentimes, like maybe out in your yard in the spring is when it's pretty common to see these popping up. But it kind of depends. All right. It depends on where you're at. So they produce these fruiting bodies, releasing the spores, and the fungi continues under the soil. Most of the time, undetected, right? You don't even recognize it's there. All right. There are some potentials for things like fairy rings, some mushrooms that are maybe growing in your yard, and you can see almost like a nice little circle of mushrooms that start popping up. And if you see that, and now you do some science, some observations, watch it over the course of a couple of years, if you still live there, right? And you will see that fairy ring gets bigger every year, all right, as it starts to progress, all right? And through all of this asexual replication going on, it's all basically the same, right? There is a fungus in Oregon, yellow honey mushroom, I think is what it's called, that they speculate that this fungus is the largest organism on the planet. Humongous. 4.5 or 5.5 square miles. And it's all the same stinking fungus, genetically identical. All right. That is awesome. All right. How many thousands of years, I could only imagine, must it have taken it to get that big? All right. Pretty impressive. All right. Okay. So, a couple of quick tables showing some of this. There are a handful of representatives that show some potentials for disease. All right. With our Ascomycota, Aspergillus is a bad type of fungus. We'll hit some details with that later. It's something that's pretty common in houses, black mold that you may see in a poorly ventilated bathroom. There are lots of potential issues for Aspergillus. We can think about some of it in the food supply as well, and may lend to some liver issues, potentially liver cancer with a bunch of Aspergillus exposure. A lot of the dermatophytes fit into this Ascomycota as well, not a lot, all of them. Trichophyton, Microsporium, and Epidermophyton. This is stuff like your ringworm and athlete's foot. They fit into this group. Our dimorphics, here's my Blastomyces dermatitidis and Histoplasma capsulatum. They're within this group as well. The Basidiomycota are mushrooms. So we can see that here, but Cryptococcus neoformans is a type of yeast that can get us. And there's some others, plant issues, rusts, and stuff like that. Amanita, the death cap, it fits into that as well, which is right here. Microsporidia, we've got these guys right here, Microsporidia, these sort of oddballs lacking mitochondria, peroxisomes, and centrioles, really kind of rudimentary, or potentially over the course of time, genetically, they lost this instead of never having it, but I feel like probably never had it. Zygomycota, here's our mucor, that bread mold. There's others in here as well. Rhizopus is also a bread mold. Other issues. So here's Trichophyton rubrum. This is one of the culprits as a dermatophyte, a skin fungi, and we can see some of the conidia and spores that have been released out, microconidium. All right, the dianoflagellates, shifting away from our fungi. These are super crazy looking, all sorts of shapes, oftentimes some sort of armor like cellulose. We've got a couple of flagella coming out of the end of it, and we think of these aquatic, oftentimes marine environments with the dianoflagellates. With the protists, or with the algae, excuse me, there are lots of variations with this and different potentials as far as photosynthesis and pigments that we can see. There's all sorts of different types of kelps, multicellular, and then we think about a lot of single cell stuff. All right. So they look like plants, but they aren't plants. All right. and some red algae over here, culprits for like some red tide situations, shellfish making you super sick if it's when some of this is blooming, if you will. All right, also another plant looking one down here, Helimedia. So plant-like, but not a true plant. All right. There are some potentials with those dianoflagellates to bioluminesce. So as I look at this image, my first thought is, wow, that's a beautiful sunset. And you just see the littlest bit of blue sky right here. That's not blue sky. All right. This is the bioluminescence from those dianoflagellates lighting up the ocean. All right. They're right here on a wave. All right. So this is the edge of the ocean as it meets the skyline right there. And that's our bioluminescence. Pretty impressive. All right. So, and I think, you know, if you were, but you know, back in the old days, sailing a ship across the ocean and you saw some of this, you might be terrified, right? What is it that's out there that's going to get me, right? All sorts of crazy sea monsters, right? And glowing water, right? Terrified. All right, the diatoms, we saw some of these before, I believe, when we were talking a little bit about some of this. All sorts of different potentials with them. And then some of the colonal algae, volvox, and some others with the chlorophyll pigments within them. Chlamydomonas is a unicellular flagellated algae. All right, and you can see the chloroplasts within there, and its little eye spot, so it can sense that. Lichens, we're wrapping it up. We're almost at the very end of this chapter. So lichens, you can often see these, you know, growing on some rocks or maybe on a tree, and varied colors. They potentially are exhibiting some oranges and greens. There are multiple interactions going on in a lichen colony, all right? So there's some fungal hyphae up toward the top. We've got some algae that are photosynthesizing. So it's like a whole community where we've got sugar producers, we've got some of the consumers tapping in the fungi, breaking stuff down, and also oftentimes some bacteria participating in these interactions as well. Although we don't have any hints of that up in here, usually there's multiple players, like three-way sort of symbiotic relationship between fungi, algae, and some bacteria. And you can definitely see lots of variation with this. As they are growing wherever, they are using all sorts of stuff, nutrients and minerals within the rock, or pulling stuff in through the atmosphere as sources of nutrients for them. So a little bit of variation, as I said, some color, orange, sometimes green, crustose, lichen. We can typically see this stuff around here. The foliose, this almost seems like we're describing bacterial colonies, right? Similar sort of descriptors. As we see this sort of leaf-like, right, foliage. So these, and this one, I always saw this like in some or flower arrangements or whatever, and I just thought, oh, that's moss. This is not moss. This is a type of lichen, all right? Different, all right? Not the same as a moss, all right? And also, as I was going through here, I'm like, pretty impressive, all right? So they used to use these lichens because there's enough poison on there that they would coat their arrowheads in some of this and make some poison, all right? Like the frogs and the, you know, poison darts that they use with those. Same but different, all right? Impressive, all right? All right. I know as soon as I hit exercise, that's the end. I do think this is a beautiful image. Any guess on who this is? Ciliate. What's the ciliate that gets people? Do you remember that? Oh man. We'll put that on the test. That's Balantidium coli. All right. Farewell. Good day. We will do chapter